Hello everyone. We are here to continue with our series on spore-forming microorganisms. We have discussed so far Clostridium perfringens, and the other microorganism is Bacillus cereus. Here we will talk about a new one, which is another species of Clostridium, which is Clostridium botulinum. Well, unfortunately, we cannot interact, but I am going to ask you a question. Have you all heard about Botox? I'm pretty sure many of you know what Botox is, or at least have heard of that. So Botox, it's a treatment that can be used to reduce the appearance of, I don't know, aging or something like that. So this Botox, and I cannot talk about how the treatment works, but all I can tell you is the Botox is the toxin from Clostridium botulinum. So they use this neurotoxin, and we're going to learn why or how that toxin works, and that might make a little bit more sense. Clostridium botulinum is a gram-positive microorganism, similar to the other two spore-forming microorganisms that we have studied. Spore-forming, we mentioned that. Now, different from Clostridium perfringens, botulinum is an obligate anaerobe, so keep this in mind. Some differences between C. perfringens and C. botulinum. So this one tolerates some oxygen, and this one is strict anaerobe. This one produces neurotoxin. This one produces enterotoxins. So these are some differences. I actually recommend that when you're studying for these microorganisms, that you can make a table. Even you can add here B. cereus, Bacillus cereus, and also describe some of those differences, and that's going to help you see visually those differences and not being confused. Okay, so it's an obligate anaerobe and it produces neurotoxins. Some other characteristics that I want to mention. I want to focus on what is most important here, because the rest you can read. You can read this. It's divided into 4 groups, and those 4 groups are classified according to the production of 7 different neurotoxins. So you will see in a slide that is coming later how each one of these groups may produce different toxins. For example, group 1 is going to produce toxin A, B, and F. Actually, I think the slides coming, there's a couple of slides here. I'm going to go back in a minute. So these 4 groups. Group 1 produce neurotoxins A, B, and C. Group 2 produces neurotoxins B, E, and E. Group 3 will produce neurotoxins C and D. Group 4 produces only neurotoxins G. And what about these different neurotoxins? So the most common toxins associated with humans are of the 7. So please do not get confused with the 4 groups and 7 neurotoxins. These are 2 different classifications. So these are 4 groups of C. botulinum, and these are 7 different neurotoxins. Okay, so neurotoxins A, B, and E have been associated with humans mainly. C and D are usually connected to animal botulism, while neurotoxin G is not. There is no evidence, or is not known what illness or which type of species associated with. Also important to know about botulism, this is the most potent toxins that we know, and this is one of the most dangerous microorganisms that we are studying here. There are high chances of death if somebody consumes a food contaminated with the botulinum toxin. The chances of death are high. It also is related to the amount of toxin that you consume as well, so we have to mention that. Okay, I already explained this, so we know that there are different groups of the microorganisms and different type of neurotoxins. These neurotoxins are heat sensitive, and we have some information later on in these slides that will be very important for you to know. What's the typical environment of Clostridium botulinum? If you see a trend between Clostridium perfringens and Clostridium Bacillus cereus, Clostridium botulinum also is found typically in soil. So one thing that is special here, Bacillus cereus was an anaerobe. Clostridium perfringens tolerates some oxygen. Clostridium botulinum is a strict or obligate anaerobe. So what that means is in the environment, you're not necessarily going to find free cells. Usually we find the spores of these microorganisms. So you can find the spores in soil, in water, GI tract of animals. It has been reported some animals carrying these spores produce, and I think produce because of the soil, because really produce is not a source of this microorganism, it's the soil. And dry ingredients also most likely contaminated from environmental sources or soil as well, and some seafood. So water and seafood are mentioned here because there's some type of this, or type E specifically, is very common in marine environments. So you will see seafood carrying this type of Clostridium botulinum as well. Now, the mechanism of pathogenicity of this microorganism has to do with the type of illness and also how the neurotoxin work. We are going to review later on in some of the slides that there are different type of illnesses. So we have wound botulinum. Let me write it down here, although it's going to come up in a few minutes. So we have wound botulism, we have infant botulism, we have the foodborne also by the botulinic toxin, and it's similar to the infant, but it's just a little difference here. So these all of these are neurotoxins, so they're going to work as toxins. They're going to work a little bit similar. So what they do mainly is that they're going to block the release of acetylcholine. Acetylcholine is a neurotransmitter that works in our neurons, and acetylcholine is going to be released by our neurons to send that message of muscle contraction. So every second while we are alive, we're contracting muscles, and that contraction is happening because the neurons are sending that message to our muscles through the acetylcholine, which is the neurotransmitter, saying it's time to contract. So this is happening every single second. The neurotoxin is pre-formed in food. Remember Clostridium perfringens? It was not pre-formed in food. You have to ingest the spores, or you ingest the vegetative cells, and they sporulate inside your GI tract. For Clostridium botulinum, the toxins are pre-formed in the food. The onset is going to vary depending on the illness. So we have here that the onset may be between 12 to 36 hours, but I just mentioned that we have different type of botulism. So the illness and also the concentration of the toxin that we ingest will determine the onset. The onset is when or how many hours it's going to take for that illness to start. So you can see the symptoms to be developed really fast if you ingest a large amount of that toxin. I was explaining the mode of action, so we can see here a little picture of the neuron. Again, there is this neurotransmitter, is called acetylcholine, that is being released to send that signal of muscle contraction. The botulinic toxin will block the release of this transmitter, so there will be no muscle contraction. So just think a little bit, what happens if you cannot contract muscles? Some of those can be the lungs, some can maybe the heart. So just think of the big consequences of not being able to contract. When you breathe, you need to contract muscles, and that's ultimately what may cause the death of the patients who have ingested this microorganism. It is estimated that the toxin may require between 24 to 72 hours to take effect. However, if you, it is based on the literature, if you consume a large amount of this toxin, the situation might be a little bit different. Okay, I have here a graph, and I will let you guys take that PDF file or PowerPoint that is uploaded in your Blackboard and read each one of these steps that is going to explain a little bit in more detail how these work, how this neurotransmitter is being blocked by the botulinum toxin. How do we inhibit or how do we control Clostridium botulinum in foods? There are different mechanisms. When we review that presentation on spore-forming microorganisms, I described a little bit of that. For the canning industry, it's very important to control this microorganism in food because canned food is considered to have an anaerobic environment, so the conditions within this type of food will be perfect for this microorganism to grow and produce toxin. So to control Clostridium botulinum, the food industry and FDA and even USDA also, they require a thermal process that is capable to control 12 logs or a reduction of 12 logs during that combination of heat and perhaps high pressures. Also, it is common in the meat industry to use nitrites. In these, if you're processing or working on a processed meat company, the use of nitrites is required for the control or inhibition of Clostridium botulinum. So nitrites are going to inhibit this microorganism. Just keep this in mind. Now, what's common in food? What type of foods may be carriers of these microorganisms? We mentioned that already. So you can have the most common in food are type A and type B and E, A, B, and E. E will be mostly common in seafood. Also, it's because we find Clostridium botulinum spores in soil. All of these products that are typically in contact with dirt, with soil, have a higher risk of become carriers of these spores. There is another interesting factor here, or an interesting fact here, which is the contamination of honey with Clostridium botulinum spores. So honey is a good carrier of this microorganism, so it can support this bacteria because of the high levels of sugar, so it's kind of perfect for the microorganism. And it has been found also a higher frequency of this bacteria, this type of product in honey. There is one specific type of botulinum that will cause infant botulism and is directly associated with honey. So in fact, there are regulations associated with production of honey to prevent the contamination with Clostridium botulinum. In terms of growth, what do we need to get this bacteria growing? We mentioned already it's an strict anaerobic, so it will grow in an anaerobic environment. That's for the vegetative cells. The temperature for growth is very wide. Now, it's these lower temperatures that I have here, 3 degrees Celsius, are not super common. So the most common temperatures are usually mesophilic temperatures, but they have found this microorganism under anaerobiosis growing, maybe slowly, but growing at this lower temperature. Now, for sure, we will inhibit this microorganism at lower pH. So pH below 4.6 and water activity below 0.85 will not support the growth of this microorganism. And because we mentioned it's a strict anaerobe, so canned food and we call MAP, which is modified atmosphere packaging, will favor the growth of these bacteria. So modified atmosphere packaging are those type of food products that are packaged with the addition of specific gases, for example CO2, and those will inhibit some type of microorganism but may not work well for Clostridium botulinum. So, and once we have also the spores, it doesn't matter what conditions you have, those spores are going to survive for many years under the most difficult and harsh conditions. Okay, we were talking earlier about different type of illnesses associated with Clostridium botulinum. So there are these 4 known illnesses. We have the foodborne intoxication, it's called, just the most common form of Clostridium botulinum intoxication. So it's just intoxication of that preformed toxin in the food. Then we have the infant botulism, which is associated with honey and is very common in children under 2 years of age. Now there is the intestinal botulism, is very rare and is associated more with immunocompromised patients, but the prevalence of this is very low. And the wound botulism, which is really not, this is the 3rd and the 4th, is really not a foodborne illness. It's not really associated with that, but also is produced by toxins from Clostridium botulinum. Now, there are differences between these illnesses. So, for example, the wound botulism may have an onset between 4 to 14 days. The infant botulism, which is associated with honey, may have a 6 to 9 hours onset. It may take up to days as well, and this one may have just hours to days, 12, 24 hours, up to even days of to have the symptoms, and it's depending on how much you're consuming. For the infant botulism, usually there is a production of this toxin in the GI tract of the infants. So the babies consume the microorganism or the toxin, both, and the microorganism is going to colonize the large intestine, and during that colonization process is going to keep producing toxin, and that's what makes it different from the foodborne botulism. The foodborne botulism is the direct ingestion of that toxin. Now, unfortunately for the infants, the problem is that the infant intestinal tract lacks of that protective bacterial flora, and maybe Clostridium inhibiting bile salt acids, and it's not going to so the microorganism can invade very easily. Now, the characteristics of that disease. We mentioned earlier that the neurotoxins will block the release of acetylcholine, which is the neurotransmitter sent by the neurons to indicate that we have to contract our muscles. So once you have ingested the toxin, the symptoms are going to start with nausea and vomiting, and then may progress to blur or double vision, will go to loss of the mouth and throat function, and this is some sort of paralysis because of that neurotransmitter is saying there is no muscle contraction here, so you lose that ability of swallow, and that makes creates panic in the person. It will cause fatigue, but that fatigue is apparently is kind of a general feeling of inability of move your muscle, and then ultimately you start losing the muscle coordination, and the most severe symptom will end up in respiratory failure, airway obstruction, and also failure of other organs because of that inability to contract. The fatality rate is about 10%, but I have mentioned several times that depends on the patient. It's going to depend on not the patient in terms of the immune system, but mainly is how much of that toxin you have consumed. And this is different from getting sick with a microorganism. When you're sick with a bacteria, so you consume that bacteria, your immune system detects that bacteria, the antigens, and will try to fight it. But when you're eating a toxin, there is no way to fight that. There might be mechanisms, I'm not familiar with those, but typically we don't have a strong correlation between the immune system and these type of illnesses. I'm going to move here to the outbreaks. Not many outbreaks here in the US. Is not a very common illness, but we have had some outbreaks. There have been outbreaks in Europe. In some countries, it's very is not very common. For example, Mexico, Central America, South America is not a common microorganism. It is more common here in the US and some countries in Europe. But still, the prevalence is very low. Unfortunately, there is a low prevalence but a high mortality rate as well. So now, in terms of detection of this microorganism, to grow this bacteria, one of the things that you need to know is no matter which media they will suggest, because different type of protocols will recommend different type of media. For example, trypticase, Burton glucose yeast extract recommended by FDA with this agar here for growth. So no matter which media, you always require strict anaerobic conditions. So an anaerobic chamber is mostly needed to grow this bacteria. Another very important factor here is that we can't work with this microorganism in a lab that is biosafety level 2. So you require a biosafety level 3 lab to work with this microorganism. And this is because of the danger that is posed by this bacteria in terms of the human health. And that's it. So if you have any questions, feel free to email me.